Okay guys, so here is our minion example for our chi-squared test, and per the request that I got over the weekend, this is the one that I'll be working, and I'll do this step by step so you guys can see it, and you guys can use this to review for your quizzes, tests, and all that, or later on when you get to stats you can see this too, if that is something that you want to do. So let's go ahead and get started. So we have a cross between two minions that are supposedly heterozygous for both glasses and hair. And this is the data that we observed. So this whole column right here is the observed data. Okay, so make sure that you have that. And if you want to write this stuff down as we identify it, that's entirely up to you. From here, the first thing we're going to do is we're going to go ahead and do our dihybrid cross. That's something that you need to do first so that you can get your ratio, because you need to calculate your expected values, because those are the most important thing right now because you don't know those. So you really can't do anything else until you start that particular step and you complete it. So if you do your dihybrid cross, and you can use whatever alleles you want, this is what you should get. You should get a dihybrid cross. I wouldn't really suggest using the multiplication rule shortcut here because it really only gives you a snapshot and the genotypes, and it's kind of hard to keep everything straight. So this is what I would do. I would do a 16 square dihybrid cross, and remember you need to FOIL out your genotypes of your two parents, and in this case they're both heterozygous because they're right here. Okay, so that means that they are going to be heterozygous for both glasses and hair. So let's say that a capital A means that they have glasses, a capital B means they have hair, and let's say that a lowercase a means they don't have glasses, and a lowercase b means that they don't have hair. So in this case, our two parents are indeed dominant for both traits, because this is just regular Mendelian inheritance, so there's a dominant and recessive. All right. So if you count up your ratios here, and remember, you really only care about phenotypic ratios. We don't really care about the genotypic ratios because we only care what the offspring looks like. We don't really care what the genotype looks like, because in our problem, if you go back to this, we're given the phenotypes, and those are the four things that we're given in our problem, because those are the things that we observed in our test. All right. So once you count this up, you should get a 9 to 3 to 3 to 1 ratio. So that's the first thing we'll do. So we'll do a 9 to 3 to 3 to 1 ratio. Okay? And if it helps you, you can also write them like this. You can write them as a fraction, because that's essentially what they are, because it's a probability. Okay? If this helps you, I would highly recommend doing it. This is the way I do it because I'm not that good at math, so I need to make sure that I keep everything straight. So out of 16 offspring, so go back to our dihybrid here, these are 16 different offspring, and you're going to see that out of 16, we would expect to get 9 that are dominant for both traits. We expect to get 3 that are dominant for at least glasses, but maybe not hair. We should see 3 that are dominant for hair but not glasses, and then we should see only 1 phenotype where they are recessive for both traits, so that little section right there. Okay, and you can check all of this using your dihybrid. That's why I would highly recommend that you use this and not use the multiplication rule. The multiplication rule is useful, but really only for solving for genotypes. And so since we need phenotypes here, this kind of just makes more sense. It's just to keep it simple and make sure that everything is organized. Okay, so now that we have our phenotypic ratios, and we know out of 16 offspring how many we need to, or how many we expect to see, we now need to see how many we should expect for our total in our experiment. So you go back to your problem here, and you take a look at the totals that you have for each and every trait, or each and every category. And we're going to have 570 that are dominant for both traits, 185 dominant for glasses, recessive for hair, 190 that are recessive for glasses and dominant for hair, and then 55 that are recessive for both traits. So what you need to do there is you just need to add all of these 4 categories up, because this is our observed test. This is our test. This is exactly what we did in our experiment, and this is what we saw. So we need to go ahead and add those up so that we can then calculate out of out of 1,000, which is our total. Let me write that here. So total offspring was 1,000. Okay. So instead of 9 out of 16, we are going to try and see how many are going to be dominant for both traits out of 1,000. So what we need to actually calculate here is we need to calculate the percentage that 9 is of 16. And now you can go ahead and set up a proportion if you would like to. That's okay. It's a little bit longer of a step. So what I'm going to use is I'm just going to use a very simple equation, and that is I'm just going to literally do 9 divided by 16, and that's going to give me my percentage in decimal form. So if I take 9 and divide it by 16, that gives me 0.5625. You can round to the nearest thousand or nearest hundredth if you would like here. I waited till the very end to round my numbers, so mine may look a little bit different than yours. It just depends on how you round. As long as it stays, in the same degrees of freedom, then we're good to go. Okay, so the next step after 9 divided by 16 is basically, this is 56% right here. This number is about 56% because we're representing our percentage in a decimal. So what we need to do is we need to multiply that times our total offspring, and that gives me how many I should expect to see. I should see 56.25%, okay, of the 1,000 offspring observed. That should come out to 562.5. So dominant for both traits, we should see this number right here. This number right here is what we should see for dominant for both traits. So let's go ahead and do that for the rest of our phenotypic ratio here. If you do 3 divided by 16 times 1,000, I'm going to skip that 1,000 step because you can do this in your calculator pretty quickly, and that should give you 187.5. And now the beautiful thing about this problem is that since our ratio is nice and clean, we can go ahead and do that for the same thing to that second 3 in our ratio, and then 1 divided by 16 equals times 1,000 gives me 62.5. So now we have our expected values. We should expect to see 562.5 in that first column. That's what we should expect to see in this first column. We should see 562.5. So we were pretty close, to be perfectly honest, in our experiment. Then for the next two, we should expect to see right around 187.5, and then the last one should be 62.5. Okay. So now that you have your expected ratios, really it's super simple. All you have to do is just plug it into the equation. Okay, and you can use this chart if you would like. Let me actually get rid of this little outcome because that's for a different problem, so you can see it a little bit better. This is what you should use if you want to keep everything nice and neat. I'm going to keep it on this page right here. I'm not necessarily going to plug it into this table. But I'll be using the same type of structure. You could also just use the formula if you would like. Now remember, you're going to have to add up each category to get your total chi-square number. So once you get this whole line filled out, you need to fill out the other the other 3, and then get your chi-square sum right here. Okay, so it's going to go right in here. All right. So what we're going to do is we're going to start with our 562.5, and we're going to take our observed, which was 570. Let me make a new slide here, and I think that'll be a little bit easier to see. So we're going to take 570. We're going to subtract our expected number. Our expected number is 562.5. So let's do that if my mouse will work here. So 562.5. And that comes out to a nice little number of 7.5. Let's do that for our next ones, which was 185 minus 187.5. Don't worry if you get a negative number here, because if you square a negative number, which is our next step in our chi-squared equation, it's going to be a positive number, so don't freak out if that's something that you don't like. Negative numbers is not something that people typically enjoy. So let's keep going. It's going to be 2.5 the other way. That one's going to be positive. And then we're going to do 55 minus 62.5. That's going to give you 7.5. All right. So now we can simply square these numbers because that's the next step in my equation. Again, if you're using the chart, you would take the difference and you would square it, okay, because that's the next step. Or if you're using the equation, you can do what I'm doing and just simply write this out. So let's do 7 point, whoa, not 8. Let's do 7.5 times 7.5. So we're going to square that, and that's going to give me 56.25. Okay. And then let's do the next one. If I square this, why do I keep typing 8? So negative 2.5 is going to be 6.25. And double check my math here because, you know, I'm not the strongest of math people. So if I do something wrong, you guys let me know. This one's not a negative, so it's just 2.5. And that one's pretty easy because it is 6.25 because we already calculated that for the other one. Let's not do that. Let's go ahead and click here. Let's do negative 7.5 is going to be 56.25. So our numbers kind of repeat here, which is kind of cool. Makes it a little bit easier. Now, the very last, or the second to last step in all of this is we are going to take our numbers that we just calculated here, and we're going to divide them by the expected. So 56.25 is going to be divided by 562.5, and that's going to give me a nice little number of 0.1. You can go ahead and round to the nearest hundredth place on this one. That's what I'm going to do. And that one actually came out to a nice even 0.1, so I don't really have to round. 6.25 divided by 187.5 is going to give me 0.03. And then down here, same thing. So it's going to be divided by 187.5, and that's going to give me again 0.03. And then finally we can do our last number here, divided by 62.5, and that gives me 0.9. Okay, now the very last step here, and if you're using this chart, what you're going to do is you're going to add up each number in this little column here. Okay, so I'm going to add 0.1, 0.03, 0.03, and 0.9 together, and that's going to give me my chi-square value. I don't really remember how to do the superscript on here, so I'm just going to use chi-square, and that equals 1.06 is what you should get if you rounded the way that I did. If yours is, you know, maybe a couple tenths below or a couple tenths above, that's fine. It just depends on how you round. So if you round earlier in the process, your number is going to be a little bit different. I think some people will get close to like 0.7 something. Some people might get closer to like 0.2 or 1.2. But the thing is as long as we can still go back to our chi-squared chart, or excuse me, our critical values chart, and get the correct assumption of our null, or characterization of our null, then we're good to go. We've done it right. So this is how you're going to be able to check and see if you've done everything correctly. So our critical values chart, you can find this pretty much anywhere. You're going to be given it on the AP exam if you do take the AP Bio exam. So it's not something that you are going to have to memorize, which is always really nice. And for this class, we're always going to use the 0.05 column because that makes sure that our statistical analysis is going to be about 95% correct. We have about 5% margin of error, and so we can go ahead and calculate our degrees of freedom over here on the left, and then come back to our 0.05 column, and that's going to be where we're going to live in this class. So in order to calculate degrees of freedom, all you have to do is you have to count up however many categories or outcomes you had in your experiment. So for our M&M lab, it was 6. For our Skittles lab, it was 5. And for this particular question, it's only 4. So you can count up your 4 possibilities. Okay? And the formula to get your degrees of freedom is you take the number of categories and subtract 1. And those are your degrees of freedom. So in this case, we had 4. So we start here. And we subtract 1 and we go up to 3. And then we move all the way over to our 0.05 column, and we are well within our degrees of freedom. So our number is way less than the 7.82 that we were allowed to have here, because our critical value is going to determine whether or not we fail to reject our null or if we reject our null hypothesis. So 7.82 is higher than our chi-square number. Our chi-square number is less than 7.82. So that means that these parents were in fact heterozygous, and this experiment is valid, and there's nothing working on the number of offspring here other than pure chance of probability. So hopefully this helps you guys. This is my first crack at something like this, recording myself. I don't really like the way I sound, so editing is going to be awesome. But hopefully this helps, and this is something that you guys can use. If you get confused on chi-square, because you will see it on your test, suggest you come back and take a look at this. There's also some walkthroughs on YouTube as well, but I mean, come on, I'm way more awesome than any of those people anyway. So keep calm, guys, and maintain homeostasis.